Hello everyone and welcome back to microbiology. This lecture is chapter 24, the digestive system infections. A brief review of the digestive system. We can follow the gastrointestinal tract from the mouth to the throat, to the esophagus, through the stomach and the intestines. There are accessory structures such as the teeth, tongue, liver, gallbladder, and pancreas. And its primary goal is to break down and absorb food molecules that pass into circulatory or lymphatic systems. With many of these systems, there exists a normal microbiota. And most parts of the digestive system are in fact heavily populated with microbes. Millions of bacteria per milliliter of saliva in your mouth exist, and about 100 billion bacteria per gram of feces in the large intestine. However, the stomach and small intestine are relatively less populated. The stomach acid in humans is very acidic, and this, along with the movement of material through small intestines, means that they have relatively few microbes. These microbes help to break down large molecules. They synthesize essential vitamins, and they outcompete transient and opportunistic pathogens, as well as have a function in regulating our mood and suppressing certain diseases. So, for diseases of the digestive system, we'll be covering bacteria in the mouth, viral, and bacterial lower digestive diseases. So let's focus on bacteria of the mouth. So while other surfaces shed cells and the microbes that may be living on them, teeth do not. Enamel is the hard mineral outer surface of the teeth, and there are hundreds of microbial species living in the mouth, but the primary one that causes tooth decay is Streptococcus mutans. Sucrose in our diet is the main culprit in tooth decay. It was uncommon until about the 17th century, and table sugar was introduced. As microbes accumulate on the teeth, they form what is known as dental plaque, which does not compose, because it is not composed of cells. Decay caused by microbial metabolism of sucrose and carbohydrates into lactic acid destroys the enamel of the teeth. And again, this is mostly caused from Streptococcus mutans. And although it can metabolize a wide range of carbohydrates, it is very good at breaking down sucrose, and it tolerates a high level of acidity. When it breaks down sucrose, it secretes dextran, which is a gummy component of plaque. Periodontal disease is a catch-all term for conditions related to degeneration of tooth support structures. Gingivitis is the infection and the inflammation of the gums, and it affects about 50% of U.S. adults. Chronic gingivitis leads to periodontitis. Well, it is chronic gingivitis, and it leads to periodontal pockets of pus, to destruction of root tips, to loosening and loss of teeth. In the image below, we can see healthy gingivae or healthy gums. At step 2, we can see gingivitis or swelling of the gums. Step 3 is where periodontal pockets begin. And step 4 is the periodontitis. Now we'll cover a select few bacterial lower digestive diseases: staphylococcal food poisoning, shigellosis, salmonellosis, typhoid fever, cholera, E. coli gastroenteritis, Campylobacteriosis, peptic ulcer disease, and other gastroenteritis. So lower digestive bacterial diseases, we can have two different forms. We can have infections, and these are caused by the ingestion of an active pathogen that multiplies in the GI tract. It's typically characterized by a delay in symptoms as that bacteria multiplies or spreads and grows. And it usually involves a fever. Then we have intoxications. Intoxications are caused from digesting bacterial toxins that therefore impair the GI tract. And these symptoms can be very quickly onset, and they can include things like diarrhea, cramps, and vomiting. And not to say that those aren't limited to infections, but we typically see those specifically with intoxications. Okay, so staphylococcal food poisoning is a common form of food intoxication, which is caused by ingestion of Staphylococcus aureus enterotoxins. The reason why it's so common is because this toxin is thermostable, so once it is formed by the bacterium, heating the food may get rid of the bacteria, but it will not get rid of the toxin itself. So if we look at the flow chart to the right, we can see that food containing protein is cooked and the bacteria is usually killed. And then the food is contaminated by the worker with Staphylococcus on their hands. And since there's no food in there or bacterium there to compete with, the Staphylococci grow quickly. Step 3, the food is left at room temperature or incubated, and the organisms in the food begin to grow long enough to release these toxins. Step 4, the food containing toxin is eaten, and then step 5, in 1 to 6 hours, a staphylococcal intoxication occurs. And as you can see, this can happen very quickly, 1 to 6 hours, so that's indicative of a intoxication. It does not require a lot of bacteria to get this done. Only about 1 million bacteria in a single gram, which is just a pinch of a lot of foods, is enough to produce the toxin to cause the disease. Refrigeration during storage is the best method of preventing toxin formation. Okay, next is shigellosis, which is bacillary dysentery. And dysentery is just the infection of the intestines, which results in severe diarrhea. And it can be with the presence of blood or mucus in the feces. The infection is caused by 4 members of the genus Shigella. It multiplies in the small intestine over about 1 to 3 day incubation. And S. sonii is the most prevalent in the US and has mild symptoms. S. dysenteriae is common in developing countries and has severe symptoms caused by Shiga toxin. Symptoms include fever, cramps, and up to 20 bowel movements per day. There are about 500,000 cases worldwide annually, and the mortality is up to 20%. This is a common source for traveler's diarrhea. And the figure to the right shows an image of Shigella on a HeLa cell, or an animal cell. So salmonellosis is the next one. It is caused by what we refer to as non-typhoidal salmonella, or S. enterica is the most common, and S. bongori. They pass through the intestinal mucosa to the cardiovascular and lymphatic systems to cause systemic disease. And this is an infection, so incubation period is about 12 to 36 hours in this case. It includes moderate fever, cramps, diarrhea, and nausea. It's estimated about 1 million cases and 380 deaths occur annually in the US. Chickens, turkeys, and reptiles are especially susceptible to infection, and it's estimated about 1 in 20,000 eggs in the US has salmonella. Where cooking thoroughly does kill the pathogen, eating raw eggs may leave you susceptible to exposure to salmonellosis. And this brings us to typhoid fever, which is caused by Salmonella enterica serovar typhi also known as S. typhi. It's spread through the fecal-oral route and is now rare in the U.S. due to sewage treatment. It multiplies in phagocytic cells that spread the pathogen through different organ systems. And after about 2 to 3 weeks of incubation, the symptoms include headache, fevers, and later diarrhea. Recovered persons can have lifelong immunity, but 1 to 3% are chronic asymptomatic carriers. And you'll see to the right here is a clipping of a news article about Typhoid Mary. So Mary Milan was an asymptomatic carrier of S. typhi, and she worked as a personal cook for many families and spread it to 51 people, killing 3. She was quarantined once, let out on the condition that she would stop working as a cook, but she refused to do so and was forcibly quarantined for the rest of her life. This was a very big issue in the U.S. court systems in order to be able to decide that legally the government had the right to quarantine someone because of the risk that she had of, or he or she has, of spreading a disease.
Checkpoint 1. What is the full name of the organism that causes typhoid?
Let's talk about cholera for a little bit. Cholera is caused by Vibrio cholerae, which is a curved rod with one flagellum. It's endemic in coastal waters, and it grows in biofilms on crustaceans. It colonizes the small intestines and releases exotoxins that causes water and electrolytes to move out of the host cell. It is one of the most serious gastrointestinal diseases. The exotoxin is simply called cholera toxin. Up to 5 gallons of fluid can be lost in 1 day, causing vicious blood, shock, and death. If untreated, mortality is up to 50%. If treated, which is usually treated with hydration therapy, and in severe cases treated with antibiotics, mortality is as low as 1%.
Checkpoint 2. After Thanksgiving dinner, the gravy sat out at room temperature for several hours before being placed in the refrigerator overnight. The next day, the gravy was recooked and then eaten as leftovers. Everyone who ate the reheated gravy, but not the freshly made gravy, became suddenly sick with diarrhea. What is the likely cause?
Okay, E. coli gastroenteritis. Most E. coli is benign and harmless to our systems for the most part. However, sometimes E. coli can pick up virulence factors from other organisms through horizontal gene transfer or lysogeny cycle of viruses, leading to induction or horizontal gene transfer of genes from other microbes. And these can confer virulence of normally harmless E. coli. They may become enteropathogenic, and that they can destroy intestinal microvilli and secrete proteins that cause diarrhea. They may be enteroinvasive, whereas they infect and multiply in intestinal epithelial cells, causing systemic illness. They may be enteroaggregative, meaning that they accumulate around epithelial cells, aggregative, and secrete diarrhea causing toxin. And they may also be enterohemorrhagic, which means that they can release the Shiga-like toxin that can cause hemorrhaging in the colon or the kidney. The reservoir for this is in domestic cattle, and 10 to 30% are carriers. E. coli gastroenteritis is the most common cause of traveler's diarrhea. The next disease of the gastrointestinal tract is Campylobacteriosis. It is caused by members of the genus Campylobacter, most commonly C. jejuni. It's a gram-negative, and it's a microaerophilic bacterium that has spirals. It is the most common digestive bacterial infection, and poultry and cattle are reservoirs. Virtually all poultry meat and 60% of milk is contaminated. This is why it's important to make sure that you cook your poultry meat and you are drinking pasteurized milk. Symptoms include fever, cramping, and diarrhea, and they usually resolve themselves within about a week. Okay, next up is peptic ulcer disease. This is caused by an interesting bacterium called Helicobacter pylori, which is a microaerophilic spiral bacterium. About 30 to 50% of the developed world is infected, and roughly 15% of them develop ulcers of the stomach and duodenum as a consequence. And the way this works is it disrupts the layer of mucosal lining that protects the stomach from its own acids and digestive enzymes. And the treatment is with antibiotics. Usually it's pretty effective. It was once thought that ulcers were caused just by, stomach acid. What's really interesting about the discovery of this peptic ulcer disease is that the scientists who postulated that it was caused by this bacterium actually drank a vial of Helicobacter pylori in order to experiment on himself. He came down with the peptic ulcer disease, and then he treated himself with antibiotics and essentially cured himself of his peptic ulcer disease. Okay, other gastroenteritis includes Yersinia gastroenteritis or yersiniosis. It's caused by Yersinia enterocolitica and Yersinia pseudotuberculosis. It exists in livestock animals as a reservoir. It's transmitted by eating undercooked meat. Next, we have Clostridium perfringens gastroenteritis, which is transmitted again through meats contaminated with animal intestinal contents. Okay, and this one has heat-resistant endospores that germinate and release exotoxins. Finally, we have Bacillus cereus gastroenteritis, which normally is a harmless species found in soil and on vegetation. It does form heat-resistant endospores, which germinate and release exotoxin.
Point 3. A rancher slaughters a pig and harvests its intestines for sausage casings. The sausage was cooked, but she still became ill with cramping, nausea, and diarrhea. What is the likely cause?
And then we have C. difficile associated diarrhea, which is thought to have caused more deaths than all other types of gastroenteritis combined. It's a normally harmless resident of the intestinal microbiome, but overgrowth after antibiotic therapy can lead to symptoms ranging from mild diarrhea to deadly inflammation of the colon. It can be treated with narrow spectrum antibiotics or a fecal transplant from a healthy donor. And this is just a fun image for you to view yourselves about C. diff infection. Moving right along, let's talk about the 3 different viral sources of diseases of the digestive system that we'll cover, which is mumps, hepatitis, and viral gastroenteritis. So mumps virus colonizes the parotid glands or the salivary glands below and in front of the ears, and this causes swelling, as you can see in this image to the right. It's transmitted in saliva and respiratory secretions. 20 to 40% of post-puberty men experience testicular inflammation, and sterility is a rare complication, but it can occur. And this mumps is protected against the MMR vaccine, another reason to vaccinate. Hepatitis is next. Hepatitis is the inflammation of the liver and is caused by one of at least 5 different viruses. So here are a list of all of the viruses. This table right here would be a good example of a mix and match question on the final exam. We have 5 different hepatitis viruses from hepatitis A through hepatitis E. With hepatitis A, the transmission is ingested. It's mostly subclinical. You have a loss of appetite, nausea, diarrhea, fever, malaise, and you can get jaundice from this. The viral load in the host is at its highest before symptoms occur, which allows the virus to spread because carriers don't appear to be sick. The incidence is greatly reduced by having the HAV vaccination. Hepatitis B, it's transmitted through the sexual, parenteral, and perinatal route. It's similar to Hep A, and yes, it can lead to liver cancer or cirrhosis. There is an HBV vaccine available in the US. There are 350 million chronic carriers worldwide, mostly in Africa and Asia. Hepatitis C is transmitted through the parenteral route. Similar to HIV, but it takes about 20 years for appearance and recognition of symptoms. And it is a chronic infection. About 85% of cases become chronic, and it kills more people in the US than HIV/AIDS, and there's no vaccine for hepatitis C. It exhibits a rapid antigenic variation that has made a vaccine development unsuccessful up until this point. Hepatitis D is transmitted through the parenteral route after chronic Hep B infection. So it does require that helper virus first. It has symptoms include severe liver damage and a high mortality rate, and there is a chronic infection to it. Lastly, hepatitis C is ingested, similar to HAV for symptoms. It is not a chronic infection, and that's about all you need to know for those viruses.
Point 4. Why is the development of a hepatitis D vaccine not a priority for the US?
Okay, so viral gastroenteritis can be caused by the rotavirus, which is a low-grade fever. You'll have diarrhea, vomiting for about 1 week. It affects roughly 3 million annually in the US with less than 100 deaths. There is a vaccine developed in 2006 which has reduced it by 98%. The next one is norovirus, which can lead to vomiting or diarrhea for 2 to 3 days. It affects over 20 million annually in the US with less than 300 deaths, and it's highly persistent on surfaces. These two viruses cause 90% of viral gastroenteritis. And to the right is an image of the rotavirus. So that wraps up today's chapter. Thank you for joining in, and I'll see you next time.